we'll be going over the differences between monosaccharides, disaccharides, polysaccharides, and giving the different examples for them. Secondly, we need to know the basic structure for these monosaccharide, disaccharide, and polysaccharide, and also the functions of the polysaccharides like glycogen, starch, and cellulose. And very important is the structure, the way it's made has a purpose for it. There's an adaptive feature for these functions, and we need to know what are these adaptive features. So let's start off with a very important thing. Pause the video and have a go at these 4 questions. For AQA biology, in this topic, they really want you to understand water potential, osmosis. So we're going to start off with like osmosis. We all know what osmosis is from GCSE. It's the movement of water molecules from a high concentration to a lower concentration through a partially permeable membrane. Now, what is water potential? And the number 4th question was about a practical that you've done in GCSE as well. So water potential is the measure of free water molecules. So it's a concentration of free water molecules, measuring that. And it's the measure of the tendency. What's the tendency of these free water molecules to diffuse, to move from one area to another area? If we increase the number of these free molecules, the rate of that movement increases. So it's basically osmosis, really. So here we have low water potential, and then here we have high water potential. Those small circles represent those free water molecules, and that big circle can represent your solute, maybe a sugar, maybe a salt. That's your solute. So water potential is very high on this side. Why? Because there is more water molecules. And water potential is lower here, less water molecules. So water will move. The net movement of water will be from high concentration to low concentration. This is very, very important mentioning that, because you will see later on how important and how exam questions are linked to the structure of these molecules are. So let's start off with describing the difference between monosaccharide, disaccharide, polysaccharides, and giving examples of each of them as well. So monosaccharides. Now the monosaccharide, mono means one. It refers to sugar molecules, which is going to be a one molecule. So individual sugar molecules are monomers, and if they are one of them, it's called monosaccharide. So if you have 2 sugar molecule, is called disaccharide, and if you have more than 2, 3 or more, is polysaccharides. So what are saccharides? Saccharides is a general term, so it refers to carbohydrates. Again, that's a general term. You have different types of carbohydrates, or I can say you have different types of saccharides. So you have glucose as a monosaccharide. You have fructose, is also a monosaccharide. You have galactose, which is also another monosaccharide. So all of these 3 different saccharides are also called carbohydrates. So it can be a bit confusing, but hopefully later on you should be able to get the flow of this. So we are talking about here monosaccharides. So I have here mannose, galactose, fructose. I have here ribose. I have deoxyribose. All of these 1, 2, 3, 4, 5 molecules, all of them, do you think they are monomers or polymers? Polymers. They're monomers by themselves. Good. Now, because they fall under sugar. So sugar does not mean glucose. Sugar is a general term. So I can say ribose is a sugar. It's not glucose, it's sugar. Deoxyribose is another sugar. Fructose is a sugar. Galactose is a sugar. Mannose is a sugar. Or glucose, which we all know is a sugar. So sugar is a general term. Okay, so we have different types of sugar like mannose, galactose. Now sugar, those sugar which are by themselves, we call them monosaccharides. So saccharide, whenever you hear the word saccharide, it always have to deal with sugars. So monosaccharides. So all of them are monosaccharides. So we have two groups, 6-carbon sugar and a 5-carbon sugar. If it's 6-carbon sugar, we call them hexose sugar, and if it's 5-carbon sugar, we call them pentose sugar. Later on in the course, you will come across ribose, you will come across deoxyribose in detail, and for now we'll be focusing more on galactose and fructose. Now that's all the introduction. So the main point for our learning point from now is glucose. Now what is glucose? Glucose is a sugar. So sugar is a general term. Now what's glucose? Glucose is a specific sugar. So glucose. I can even say glucose is a monosaccharide or disaccharide. Monosaccharide is by itself. Why saccharide is a sugar? Glucose is a monomer. That's being too general now. So glucose is there. So it's a hexose sugar. It's got 6 carbons. Now glucose can exist in 2 forms, and that's why we are going through this. And you have to know these 2 forms really, really well, to an extent that you have to draw this in your exam from scratch. I have here one glucose there, another glucose there. They're very similar and they are isomers. So alpha glucose, can you see that carbon there? It's got OH attached to it, and the OH is pointing downwards. And if you go to beta glucose, the first carbon there, the OH is pointing upwards. For the exam, you have to draw this. So let me go through this. The carbon there, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. This is called hexose sugar. You have to be able to draw this. Remember, there's oxygen in between linking that 2 carbon there, and you have to draw these from scratch. So practice drawing this. You can think of the OH. The OH is pointing downwards. So some people say it's like a fish. Fish always goes downwards. And beta for bird. OH is upward, like the bird's going up. That can help. So remember I told you about sugars. Sugars are very general. Now remember I said monosaccharide. Saccharide refers to sugar. Anything linked to carbohydrates or sugar is called saccharide. Now monosaccharide means sugar by itself. So I have alpha glucose, which we have done, so we're going over beta glucose. We also have other one, fructose and galactose. So alpha glucose is commonly used, though it's the main substrate we use for respiration in our mitochondria. It can also be used if they are joined together to form a polymer. It can form starch or glycogen, which we'll do later on. Beta glucose is the isomer, remember? Now beta always links to plants. So when they join together, they form a polysaccharide. Now we don't have that in our body. It's found in plants, which makes the cellulose, the cell wall. That will be going over later on. Fructose is another sugar, monosaccharide, that's found in many fruits, and it's sweeter than glucose. And can be found in many fruits and found in nectar. Galactose is another monosaccharide, another sugar, and it joins with the glucose to form a lactose, which we will do later on. So in this slide, just showing you the different types of monosaccharides or different types of monosugars. Now, so when we have 2 monomers, 2 monosaccharides, if you have one monosaccharide here, another monosaccharide together. So I have here one glucose, that's a monosaccharide, another glucose there, another monosaccharide. When these 2 saccharides, when they react together, they release water, and that's called condensation reaction. When they release water, these 2 monosaccharide will now be chemically joined, and that's a covalent bond. Now I have 2 monomers, I have 2 monosaccharide, so this whole molecule will be called generally disaccharide. So disaccharide means 2 sugar together, but that's a very general term. I can have any 2 sugar joined together, that's called disaccharide. But what's maltose? Maltose is the specific name for that disaccharide. So I have here a alpha glucose, and I have another alpha glucose. I have two glucose together. So when I have two glucose joined together, it's called a maltose. So maltose is the specific name, but if I was to go broad, it falls under the group of disaccharide, two sugars together. Now I can break down maltose. It's the opposite reaction, as you can see, adding in water to hydrolyze that bond. Now the bond between them is important as well. For the exam, you should be comfortable in drawing a maltose and drawing that chemical bond. That chemical bond we'll do later on. It's called a glycosidic bond. Okay, so focusing here is on the glycosidic bond. I have a disaccharide being formed. I'm talking about specifically maltose. So alpha glucose. I know it's alpha, the which is pointing downwards. When these two react, water molecule is removed, and that bond there, that's glycosidic bond. Don't skip this part, don't rush it. You need to draw it over many times until you're perfect in identifying and drawing from scratch how a glycosidic bond looks like and how these two molecules looks like as a disaccharide. Don't forget that water always released out from a condensation reaction. So in this example, I have said if two glucose joined together, it's called a maltose disaccharide. How about if I was to use different types of monosaccharide, different types of single sugars? So if I have two glucose together, it's maltose. We've done that already. Glucose and fructose, it forms a disaccharide, and that is called sucrose. Glucose and galactose forms lactose. So you need to know these names: lactose, sucrose, maltose, and what are the individual monomers that makes them for your exam. Now, don't forget these three reactions which forms these three different disaccharide, all of them release water. Why does it release water? It's a condensation reaction. Now, what's a polysaccharide? So poly obviously means a lot of them. What's saccharide? So sugars. So lots of sugars together. Obviously, it can't be one sugar. That's monosaccharide. And it can't be two sugar. That's disaccharide. So it has to be more than two. So three or more. Or you can say more than two, however you say it. So polysaccharide is that you have to have more than two, not two, more than two. So in here I'm having lots of sugar molecules, and I have here 1, 2, 3, 4, 5. So that's well above 2. So these are joined by glycosidic bond. That's a polysaccharide. Polysaccharide meaning lots of sugar molecules joined together. For the exam, you should be comfortable in drawing a polysaccharide like this, which are made from alpha glucose. So as you can see here, these are a glycosidic bond, and these are your individual sugars. Right, so looking at this structure, this molecule here, what do you think this is? I have a glucose here, I have a glucose here. So it's a condensation reaction, and I'm having two molecules together. It's sugars, so it's disaccharide. Maltose is the specific name, and water being expelled out. Now, the bond between them, when you see these two bonds, that's a covalent bond between these two glucose molecule. This bond is called 1,4 glycosidic bond. So generally, that chemical bond is called glycosidic bond. What's 1,4? 1 refers to the carbon number 1 of this molecule and carbon number 4 of the other molecule. And that's why it's called 1,4 glycosidic bond. And you have to know that. If it's in a straight chain, all the glycosidic bond will be 1,4 between carbon number 1 of one molecule and carbon number 4 with the other molecule. What's the condition? It has to be a straight line. They can't be branching out, has to be a straight line. If they are branching out, okay, so in here, as you can see, they go in a straight line there. So if they are in a straight line, all of these glycosidic bonds are going to be 1,4, 1,4. Look at that one there. It's not a straight, it's branching out, right, from the norm. So that bond between these two sugars is still a glycosidic bond, but it's between carbon number 1 of this one and carbon number 6 of that one. So it's 1,6 glycosidic bond. So the moment they branch out, is that branching point, it will be 1,6 glycosidic bond.